Okay, folks, so we're going to continue with our discussion. We're going to look at our results. And, folks, I just want to let you know that when you turn in this second Lab Exam 2 Homework Study Guide packet, I will be looking for sketches of your results. And you can put them on the homework itself, you guys, or if you want to add pages, you can do that, right, for your diagrams. Okay. So, you guys, so first of all, let's take a look at our Chapter 9 motility, and we inoculated Motility Auger deep demos. Now, let me see if I can find those. I forgot to grab them. And, folks, you'll remember that the Motility deep auger, it has a really low concentration of auger in it. And so, do you remember how we inoculated these, or like how you would inoculate these? How would you inoculate a Motility Auger deep? Would you use your loop? No, your needle, right. Okay, so you're going to stab your microbes through the auger. And remember that with such a low auger concentration, it's like 0.4% weight per volume. That's 0.4 grams per 100 mils, right? If the bacteria are motile, they'll be able to swim away from the stab line and move into the surrounding auger. Now, why would they want to do that? Why is that an advantage? To find food, right? Because if you're growing along the stab line, you're going to use up all the food, and also it's accumulating waste products, right? So it's a huge advantage if you can move away from the area where there's a lot of waste and move away from an area where there's not much food to find areas that do have food. So you guys, in researching the formula for the Motility Auger deep, I discovered that it's not good for growing fastidious microbes. So what are fastidious microbes? Microbes that require a lot of preformed organic molecules, a lot of growth factors. And unfortunately, and I didn't know this, our atrichous non-motile microbe is Streptococcus lactis, and Streptococcus, the genus Streptococcus, they often require a lot of growth factors. So ideally, had we had a non-motile microbe that grew, we would have had a single red line straight down and right back up again, showing that the microbes are only growing along the stab line. But in this case, the Streptococcus, they didn't grow at all, right? There's no red here. So again, Gesundheit, on the lab exam, what we'll do for this particular topic, we'd have two photographs. One with a non-motile microbe, so you'd see just the red line along the stab pathway. And then the other one, this guy did turn out well, you guys. This is Proteus Mirabilis. This is a cousin of E. coli. And just like E. coli, Proteus is covered with flagella. What's that flagellar arrangement called when the entire surface is covered with flagella? This is what you want to practice for the lab exam. Do you remember those? And you guys can look at your motility handout. That's totally fair. So if the whole surface is covered with flagella, what's that flagellar arrangement? Good, peritrichous, good. Yeah, no, that's good, you guys. Using your handouts is awesome, right? Okay. And so, indeed, you guys, the members of the genus Proteus, they are so motile. If you inoculate an auger plate with Proteus and incubate it, by the next day, they call it swarming. They would have swarmed over the entire surface of the plate. It's like a frosting of bacteria. So they're really challenging to get isolated colonies. But with regard, you guys, to the Motility Auger, what I would suggest, if you were actually running this test, you hold it up to the light. And, of course, you're going to have nice red color along the stab line where the microbes are growing. But what you're looking for is diffusion of the red into the surrounding media. Because remember, wherever the red is, the bacteria are. And so by holding it up to the light, you can see that the bacteria are moving away from the stab line into the auger itself. So would this be positive or negative for motility? Positive, right. Good job, you guys. And most likely the structure used for motility is what? Flagella. Awesome, you guys. And this is so cool, you guys. I see a little gas bubble there. So Proteus was fermenting, making gas while it was being motile. That's kind of cool. Okay. So again, folks, we'd have photographs on the lab practical to ask you to interpret the Motility Auger deeps. Okay, so you guys okay with that one? Okay. All right. And so, folks, what I'll try to gonna try to kind of flip back and forth between the lab study guide packet just to make sure we're covering all of the questions, okay? So you guys, we've already answered the first question, what is motility? And folks, number 2 got deleted. Number 3 is asking you to compare and contrast prokaryotic flagella to eukaryotic flagella. And you guys, I'm gonna assign that as homework. So you could just do a computer search. You could look in, if you have your a micro textbook, and just look up flagella, you know, bacteria, eukaryotic flagella. They'll tell you the difference. So I'm going to assign that as homework, okay? And you're going to draw a cartoon comparing and contrasting the prokaryotic bacterial flagellum to the eukaryotic one, and do label them, you guys. So that's I'm checked for labels when I'm scoring your homework packets. Okay, and then folks, you'll see on a lot of these questions, I actually supplied the answers. Right? And remember, you guys, this is going to be a great source of topics for the lab exam. So try to start studying as we go so you aren't leaving it to the last minute. Okay, so we're talking about the components of the bacterial flagella. Talking about the serotypes, the E. coli O157H7, and it gives you an idea of the significance. And then, folks, another homework question there is on endoflagella and axial filaments of bacterial spirochetes, Treponema pallidum, Borrelia burgdorferi. And again, folks, you might recall from lecture when we were talking about bacterial structures, we talked about endoflagella and axial filaments. So your lecture notes would also be a source to answer those questions. Okay, we'll turn the page, you guys. And number 6 is just describing the flagella arrangements, which we did yesterday and which are also in your motility handout. And we also answered, you guys, number 8, Brownian motion. Is it true motility? Nope. Good. And you guys, then number 9, it says what is taxis, negative and positive taxis, chemotaxis, and phototaxis. Again, we went over those answers yesterday. Not yesterday, Monday, right? Okay, so check your notes there. And number 10, you guys, is just describing the use and interpretation of the motility auger deeps. Okay, so I think we've hit most of the key topics on motility that we would ask you on the lab exam too. So you guys have any questions on those? Pretty straightforward. All right. So then, folks, what are we going to do next? We're going to move to Chapter 11, Aseptic Transfer. And you guys, which microbe did you transfer to your TSA big butt slants? Which was the microbe? E. coli. Good. Okay. So let's first, before we go into the questions there, let's first talk about possible results on the TSA big butt slants. How did you inoculate your TSA big butt slant? Needle, and you did a what? A stab. Why did you stab the auger, you guys? Why did you stab the butt? Good, that's right. You're testing to see if your microbe can grow anaerobically, so the butt is going to be anaerobic. And then what did you do after you stabbed the butt? You streak the surface. And why did you stab and streak? Why did you streak the surface of the slant? Perfect, you guys, to see if your microbe can grow aerobically. Okay, good. All right, folks, so let's just walk through some of these questions. So what you want to do is hold your TSA butt up to the light. And folks, always move your labels. You don't want your labels to interfere with your observation. So like if you had your label here, just move it up to the top, okay? So what you want to do is tighten the cap, you guys, so nothing leaks out. Hold it up to the light. And one way, you guys, to tell is if your E. coli grew in the butt, you're going to see cracks and bubbles. Do you see cracks and bubbles in there? Do you see cracks and bubbles? Yeah. Oh, you guys, so right now we're just doing observations now, and then we'll go back and answer the questions, okay? So you guys, on your TSA big butt slants, if you hold it up to the light, do you see cracks and bubbles in there? Yes. Yeah, awesome. What do you think the cracks and bubbles are from? Fermentation. Your E. coli is growing anaerobically using fermentation and producing what? Gases, right? And the gases you guys classically produce during fermentation are molecular hydrogen and or carbon dioxide. So that's what's causing the cracks and bubbles in the butt, right? Is your E. coli growing anaerobically using fermentation, producing gases like molecular hydrogen and CO2, right? So is that evidence that your microbe grew? Yeah, and it's also evidence that your microbe carries out which process? Fermentation, right, with gas production. What could the gas or gases be?
Hydrogen or carbon.
Yeah, molecular hydrogen or CO2. Awesome, you guys. And we'll see later when we do some of our metabolic tests. One of the metabolic tests involve inoculating a big butt, what's called TSI auger. You'll see the microbes, some of them make so much gas, the auger literally explodes, right? And so one thing you guys, I think I forgot to mention is for safety's sake, whenever we're doing big butt slants, we should always put a paper towel in the bottom of our can and spray it with disinfectant. And then we should always, for today it wasn't an issue, guys, but when we start doing the TSI big butt slants, I'll also have you spray the tubes with disinfectant before you handle them, and you'll always handle them with gloves. And the reason is, you guys, again, with the TSI, there's so much sugar, there's so much gas, is the auger blows apart. There's so much pressure, it forces the auger out beneath that loose cap, and you actually have microbes and auger on the outside of your slants. It's pretty impressive, right? But we want to make sure we keep you all safe. Good job, you guys. Okay, so the cracks and bubbles are evidence of growth, evidence of fermentation with gas production. Okay, you guys, how would you determine if you have growth aerobically? What would you look at? Which part of it are you going to look at? The surface of the slant. And did all of you get aerobic growth? Good job. And remember, you guys, E. coli is a facultative anaerobe. It uses what if oxygen is present? Which process to make ATP is it using? Aerobic respiration. Good, you guys. And so it's going to make maximal ATP using aerobic respiration. It's going to make less ATP if it's carrying out anaerobic metabolism, either fermentation or anaerobic respiration. Good job, you guys. Can you, and we're just kind of learning this, you guys, looking at your big butt slant, can you tell if it's pure or contaminated? It's tough, you guys. If the growth on the surface looks the same, right? It looks like the same kind of frosting. That's hopeful that you didn't get contamination. You guys, what if you saw like the kind of non-pigmented colonies and maybe there's yellow or orange colonies in there? What would that suggest? Contamination, right? And how you guys might your culture become contaminated? What kind of mistakes might we have made? Yeah, right off the bat, right? We might have forgotten to sterilize our needle. Or you guys, what if I sterilize my needle and accidentally I knocked it into, I don't know, the bench top or something? Could that have caused contamination? Yeah, right? Anything the needle touches after you've sterilized it, you presume it's been contaminated. Okay, is it possible, maybe you forgot to wear your gloves and maybe you touched your finger to the lips or something? It's possible you had transfer of skin microbes to the tube. Yeah, that's a possibility. Good. Okay, good. All right. Oh, and another thing, you guys, with contamination, you always have to ask yourself, was the medium contaminated, right? That's something kind of out of your control. But if you're seeing you're getting a lot of contamination in your cultures, you want to say, is the autoclave not working right? Yeah? Maybe somebody forgot to autoclave the medium, right? So you're always kind of thinking, how could this have occurred? How could the contamination have occurred? Now, you guys, the opposite. What if there was no growth? What we call a failure of transfer. Can you explain some reasons why nobody grew in a TSA big butt slant? What are some reasons that we might have gotten a failure of transfer? The needle was too hot. Good. You fried your little babies. Yeah. What else? What if, you're going to laugh at this, you guys, what if you missed the microbes? Remember how we gave you the E. coli plates? Right? What if you harvested cells from auger where the E. coli weren't? You literally missed the cells. Is that a possibility? Yeah, probably not a great possibility. What if the pure culture we gave you, what if the bacteria were dead? Is that a possibility, right? And that you have no control over that, right? And then another thing, folks, is the media that you've transferred the bacteria to. Maybe your bacterium can't grow on that medium, or maybe you grew them at a temperature at which they cannot grow, right? So these are all possibilities for failure of transfers. We try to make sure, you guys, for the microbes we give you, that they can grow on the medium, and that the temperature at which we ask you to incubate, we try to always make sure that that temperature, your microbes will grow really well at that temperature. Okay? Alrighty then. So you guys do make sure to include in your lab homework study guide packet a drawing of your big butt slant, labeling growth on the slant, labeling growth, gas bubbles, cracks in the butt. Okay. And again, you guys, you can squeeze it in somewhere, and I apologize because I didn't leave you space for it. But if you want, you can do your diagrams, say, at the bottom of a page, or what a lot of people like to do is just use a separate page to do their diagrams, and then they just staple it to the packet. And it's fine, you guys. You can just staple any additional pages. You can just staple it to the back of the packet. Okay? All right, guys, so let's take a look at then, let's see, 1, 2, 3, 4, 5, 6. On page 7, let's just make sure that we've hit most of the topics here. So you guys, the first one there is describing growth of bacteria in broth. So we've got some of the old urine cultures here, folks, so you can come up here to look at them. But there's 3 indicators of growth in broth and liquid medium, folks. And first of all, we all really comfortable. It appeared that this broth has microbes in it. Yeah, and how can you tell? It's turbid, it's cloudy, right? So we know cells are present. Do we know they're alive or dead? We don't really know? Yeah, and very importantly, guys, can you tell just looking at this with your naked eye, can you tell if it's pure or mixed? Can't, right? That's a big disadvantage of broth cultures. Good. And then, folks, if you have a chance, you can come up here, tighten the cap here, because what you want to do is hold it up to the light and look at the bottom of the tube, and you can see a collection of cells at the bottom. That's referred to as a button or a pellet. And the reason I wanted to describe that is in some broth cultures, all the cells settle to the bottom, so that when you pull them out of the incubator, the broth appears transparent. And you think, oh no, I had a failure of transfer, right? My microbes didn't grow. But before you give up, always take a look at the bottom and always just gently mix. And you might see, oh, I do have really good growth. They just all settle to the bottom, okay? So a button of cells, right, at the bottom is also an indicator that cells are present, that you had growth. The last thing, folks, and these little guys didn't do it, but some bacteria, especially if they're strict or obligate aerobes, they can form a type of biofilm at the air-broth interface. And that layer of cells is called a pellicle. It's called a pellicle, and it makes sense. It's a survival advantage. If you can use aerobic respiration, and you're living in the broth, where will the highest concentration of oxygen be? It's right at the air-broth interface, right? So we're going to have high O2 up here, and then you guys, oxygen does not diffuse well through water-rich environment, so we're going to get a concentration gradient. So the deeper we go in the broth, the lower the O2 concentration. So again, if you're a microbe that can use aerobic respiration, if you can form that pellicle at the top, that's going to be the best place for you to grow because you can make the most ATP there. Does that make sense, folks? Okay, let's see here. So you guys, again, we're on aseptic technique and transfers and chapter 13 isolation. So folks, how can you determine if microbes are growing in a liquid culture? The turbidity, right? And also we're going to check for a button, right? Okay, good. And then, folks, number 3 is to describe the steps within aseptic transfer. And what I want you to think about is when, during that process, your culture could get contaminated. Okay, think about where along the way could I have contaminated my culture. And you guys, maybe that could be a possible lab exam station. We'll have maybe 2 or 3 either plates or slants, and we'll ask you if you think they're pure or contaminated. And if one of them's contaminated, we'll ask you to explain how may it have become contaminated, right? And we'll usually just ask for one or two possible explanations, right? Okay. And folks, we already did 4, what might cause a failure of transfer, okay? And then number 5, you guys, by just looking at a broth culture, can we determine if it's become contaminated? Nope. And then, folks, number 6, I don't think I explained this very well. If you have a broth culture, right, and you want to, you hope it's pure, but you're not sure, can you name a couple things you could do to try to determine if it's pure or mixed? What's something that you could do within about 10 minutes? Maybe gram stain it, right? Right, maybe gram stain it. The only problem is, you guys, sometimes different bacteria will appear the same in a gram stain, right? Another possibility is you could take a loop full of broth and streak it on a streak plate and see if you get different kinds of colonies, right? Different kinds of colonies forming, because our hypothesis is different microbes will form different types of colonies. And again, you guys, there's always a drawback. Sometimes different bacteria will form similar colonies. But at least initially, you guys, gram staining or streaking an auger plate would be 2 ways to help you determine if your culture was still pure or if it had become contaminated. And then, folks, 7, we're moving into the chapter 13, isolation of bacteria using an auger plate. So, you guys, let's just talk about our observations first. So, first of all, you guys, did you get growth? Did your bacteria grow? Did you have bacteria in the urine? Remember, we're checking for a bacterial pathogen causing cystitis. So you guys, are there microbes? Were there microbes in your urine? Yeah, and how did you tell? You have colonies growing. Good job, you guys. And then, if there was no growth, can you provide a couple of explanations? Again, you guys can think failure of transfer. Maybe your loop was too hot, right? Maybe you're growing it on the wrong medium, for example. Another thing might be, folks, maybe the urine was stored improperly. You know, maybe it got really hot and the microbes died. So you always want to ask about how was the sample treated before you got it. Good. Okay, guys, so let's ask this. Do you think you have a pure culture from your urine, or is it a mixed culture? And what I'm going to suggest you guys, this can be tricky sometimes. Hold it up to the light and look at all 4 quadrants and see if you can detect only a single type of colony or if you think there's 2 or more different types of colonies. And again, folks, it's really important. If you have tape labels on the bottom, it's really important that the tape doesn't cover up colonies. So what I would suggest is remove the tape from the bottom and put it along the side of your plate, because you want to be able to hold up the plate to the light. You want to look at it from the bottom of the plate, and you want to flip it over and look at it from the top of the plate. Okay? And again, folks, sometimes there's a second microbe hiding in quadrant 1 or quadrant 2. Okay? So what do you think, you guys? Do you think those urine samples, were they pure cultures or mixed? Awesome. Good job, you guys. That's awesome. Okay. So, let me ask you, how did you determine? You're right, but how did you determine they're mixed? Different types of colonies. Great, you guys. That is so awesome. Okay, so you guys, all of you should have had a common commensal of your skin called Micrococcus luteus. Micrococcus luteus, these are gram-positive cocci. They often form tetrads, little packets of 4 cells, if we were to gram stain them. And folks, what's really cool about them is at low temperature, they usually make a yellow pigment. So do you have any yellow colonies on your plates? Can I just take a peek really quick, you guys? Just look at somebody's plates really quick. Okay. Oh, there it is. There's a Micrococcus luteus, a little yellow guy. And the little guys here are Micrococcus. Yeah, so they form these little yellow colonies. Awesome. So you guys, if we were screening the urine to see what's the pathogen, Micrococcus luteus, I think that's just part of the skin, normal microbiome, maybe on the mucous membrane. So I wouldn't think that the Micrococcus is the pathogen, right? But some of you have a much bigger non-pigmented colony. Who has that one? Who has the big non-pigmented colonies? Okay, was that Mrs. Pink or Mrs. Blue? That was Miss Pink. Okay, so you guys, those great big colonies, those are E. coli. And they are a big suspect for causing bacterial cystitis. And remember, you guys, how we were talking about in women, we're not engineered that great, in that the opening of the urethra is really close to the opening of the anus, right? So whenever we defecate, our feces has all those fecal bacteria present, including E. coli, right? And so maybe when we use the toilet tissue to clean ourselves, maybe accidentally, we wipe some of those E. coli close to the urethra. And remember, you guys, E. coli have what? Flagella, right? So they can cause an ascending infection, and very often they're responsible for bacterial cystitis. So Ms. Pink, I would be a little bit worried that that could be the cause of the bacterial cystitis, all right? And what we would do, you guys, this is one of the points. So folks, we'll do a follow-up here. So folks, did you get isolated colonies? Did you get isolated colonies? It doesn't matter where. Does not matter where. Did you get isolated colonies somewhere on the plate? Then you succeeded, right? You guys, it doesn't matter where the isolated colonies are, right? If you have lots and lots of bacteria in your sample, you might not get isolated colonies until quadrant 4. But if there's not many bacteria in your sample, you might not get, if there's not very many bacteria in your sample, you guys, you might get isolated colonies in quadrant 1 or quadrant 2. In quadrants 3 and 4, there'll be nothing, right? That's nothing you did wrong. It just shows that there weren't many bacteria present in your sample, okay? So you guys, what's the big deal about an isolated colony? Is an isolated colony, you guys, a colony not touching another one, do we consider it a pure culture?
Yes.
Yeah, because our hypothesis is that all the cells, so I'll do a colony here, you guys. The hypothesis is that all the cells in that colony descended from one bacterium, right, or one fungus that landed on your plate. And you guys, in one of those visible colonies, you can have 10 to the 8th to 10 to the 9th cells in a visible colony, right? So an isolated colony is a pure culture. So you guys, what if, let's say, I'm just going to pretend, you guys, let's say these little guys are the Micrococcus. Those little tiny colonies. I don't have yellow ink, right? And you're not thinking, I don't think that's a pathogen, but those great big colonies, you guys, the great big non-pigmented, that's an E. coli. How can you get your potential pathogen, your E. coli, into pure culture and pretend you got lots of TSA plates? That's perfect. That's right, you guys. So you're going to get another plate, right? And using aseptic technique, you're going to transfer cells from the E. coli colony to a brand new plate. We call that a restreak, right? So you get a brand new plate, and you're going to restreak cells from the E. coli colony to your new plate, right? Incubate. And then in 24 hours, you should have a streak plate that only has E. coli growing on it. Now, why is that a big deal? Because we want to do metabolic tests on it to identify it, make sure it's E. coli, and what else might we want to do? Do antibiotic sensitivity tests on it, right, to make sure we're giving the right antibiotic. And we're going to be doing that next week, you guys. All right, so folks, do you see how with streak plates you can go from a mixed culture to pure cultures? Okay, it's a very powerful, really powerful technique. Good. All right, you guys. Then, folks, those of you that have Miss Blue, do you have the small yellow Micrococcus colonies? And then who else do you have? Or what else do you have in there? Big red, kind of brick red colonies. That's a cousin of E. coli called Serratia marcescens. And you guys, I'm gonna actually do this myself because I'm not sure how commonly Serratia marcescens would cause a bladder infection, if ever. Okay, so I'll look this weekend. I'll do a little bit of research to see if it's would be high on our list of pathogens, or if it's potentially a contaminant, right? Serratia. It's so funny, you guys. Serratia. It was originally described as causing diseases, infections of plants. And this is so bizarre, you guys. Back in the days of when the U.S. was more concerned about biological attacks, They did this study, you guys. This is horrible. I'm smiling. I shouldn't be smiling. But what they did is they thought Serratia marcescens wasn't a pathogen of humans. They would spray. They had an experiment where they would spray it from airplanes over San Francisco to see where it would float to, right? And then they would sample for it. It's great because it's got those nice big red colonies. But they found since then really, really highly immunocompromised patients, like your HIV/AIDS patients, it can cause infections, right? So that's not a good idea to go around spraying Serratia marcescens over your population. It's not a good idea. Okay, but you guys, we'll do homework to see if Serratia marcescens in an older person, and you guys, let's pretend that these patients are 70 or 80 years old. Let's see if Serratia could actually cause cystitis. Okay, that'd be kind of interesting to find out. Okay. And again, folks, when you start doing your unknowns, make sure when you streak your plates, you hold it up to the light and look to see if you have contamination in quadrants 1 or 2. Sometimes your contaminant is in low numbers, so you're only going to see it in quadrant 1 or 2, right? You might not see it in quadrants 3 or 4. So always carefully inspect all 4 quadrants. Do you think we could do that on the lab exam, folks? Do you think we could give you some streak plates? Say 3 different streak plates, and say which ones, if any, are contaminated. And do you think we try to be sneaky and have one of the plates where the contaminants are only in quadrant 1, so you'd have to carefully inspect them? Yeah. Does that make sense, folks? Okay. All righty then. Okay, guys, so let's take a look then at our lab homework study guide packet. And you guys, number 7 is asking how do you streak the plate to get isolated colonies. And then, folks, it's the second part is starting with the mixed culture. How are streak plates used to obtain pure cultures? So it's just that the challenge that we already talked about. Say in your urine cultures, you want to isolate that bigger colony, right? In pure culture to do metabolic tests or antibiotic sensitivity tests. You'd get a fresh TSA plate, use aseptic technique, harvest cells from the colony that you're interested in and restreak to a new plate, and in 24 hours you'll have pure culture. And then you do your metabolic test, and then you do your antibiotic sensitivity testing. Good job, you guys. All right, so let's go over here to the last couple of questions, you guys. So you guys, genetically, how are cells in an isolated colony, how are they related to one another? They should be like genetic clones, right? Now, we know there's mutations in there, right? Because every time DNA polymerase copies the bacterial chromosome, it does make some mistakes. But we say, quote unquote, they're genetic clones of one another, right? And thus it's a pure culture. Good. And then you guys, what are pure cultures? Single type of microbe, right? And why are they such a big deal in medical microbiology? Yeah, to do our metabolic test to identify them. And what else? If it's a pathogen, antibiotic sensitivity testing. And again, you guys, we're going to do antibiotic sensitivity testing next week. Good. All right. Okay, you guys. So, just because I will forget, folks, that chapter 4, this is a practice quiz on control of growth. Control of growth, you guys, it's a huge topic. It's a lot of information. And so I included this practice quiz or exam so that when you're studying, you can study really efficiently, so you can see these are the topics I need to know for the lab exam. So you might want to just skim them, you guys, before next week when we actually start doing the exercises. So when we discuss or you're doing a procedure, like, oh, I remember that was one of the quiz questions, right? It'll be more meaningful for you. Okay? All right, folks, so we just have one more thing to do, and that is we want to do a follow-up on those incredible airborne plates. Are you guys blown away by the growth on your airborne plates? Do you all have your airborne plates? You guys, I see there's plates still over there. So don't hesitate. You want to go over and grab your team airborne plates because we're going to discuss the data we're going to collect. Okay? And again, folks, I apologize because I haven't quite got this study guide together. I didn't leave a place. I don't think I left a place for you to record the data from your airborne plates. So again, folks, I'd like you to record 2 tables that we'll discuss. And again, you can squeeze this in. Maybe at the bottom, let's see here, this is, what page is that? 1, 2, 3. At the bottom of page 3, you guys, you could squeeze these 2 tables in, or if you just want to put the 2 tables with your data on a separate sheet and just staple it to the lab packet, that's perfect, right? But I will be looking for these 2 tables. Okay, folks, so first of all, let me give you the results on our airborne plates. What are the 0-minute plates? Why did we do 0-minute exposure plates? Those are negative controls, right? Because you guys, we need to make sure that the auger in the plates was sterile, right? If it was already contaminated, the whole experiment is a bust. So we ran the negative controls, you guys, the 0 minutes. And again, we always want to remove the tape label from the bottom so we can see. Often the tape hides colonies. So you guys, this is the TSA 0 minutes. So does it look pretty sterile to you? Yeah. Okay, so you guys, the TSA 0-minute control, no growth, right? So you can put 0 colonies or NG for no growth. That's good, right? We don't want anything growing on our negative control. And then, folks, here's the Spirochetes dextrose auger, 0-minute negative control. And let's see if it's looking sterile. See if our autoclave is working. And folks, what do you think? This is the Spirochetes 0-minute exposure, negative control. Is it looking pretty good? Looking good. No growth, 0 colonies, you guys. So both of our negative controls are looking good. All right, so we're going to put 0 minutes, so 0 colonies, 0 colonies. So this first table, you guys, is for total number of colonies. Now this can get tricky because you guys have so much growth. So let's presume, we'll use this one, you guys. This is from the Tuesday, Thursday group. This is their TSA 60-minute plate. Now a key to this is we don't open the plates, you guys. Oh my God, there's so many fungi in here, and they've made billions of spores, so we're never going to open the plates. But what we want to do, you guys, is peel back the tape from the bottom, because again, the tape will hide the presence of a lot of your colonies. And it's important we count all the colonies, right? No matter how little, no matter how big, each colony counts. We're counting total number of colonies, okay? So again, folks, what I would do, I like to look from the bottom of the plate, because sometimes one colony will grow over another colony, and you wouldn't see it looking down from the top. So what I like to do, you guys, is look from the bottom. I'm going to hold it up to the light, and maybe what you want to do is take a pen or wax pencil and put a little dot by each of the colonies as you count them, and that way you'll know that you didn't double count or you didn't miss somebody. And so each little colony, you guys, counts no matter how big or small. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23, 23. So you guys, on this plate, I'm just making this up, I'm counting, oops, I missed some, 23, 24, 25. Okay, on this plate, my total number of colonies, TSA 60 minutes was 25. This, you guys, yours is going to be different, and you know why? Different places in the lab have different loads of microbes in the air, right? So yours might be really different. But then again, folks, right, we're first counting total number of colonies, right? And the guess would be, you guys, if at 60 minutes I have 25 colonies, what would be your guess? The number I should have in half the time?
Half of that.
Yeah, half of that. We would guess. Now, it doesn't always work, right? But I'm going to make this data up, you guys. I would guess I probably have around 12 colonies at 30 minutes. Okay? And then, folks, if I took my SAB plates, do you think I should have more colonies on SAB or fewer colonies on SAB? Now, let's think of it, you guys. So TSA, what kind of functional media is it? It's all-purpose. It's going to grow both bacteria and fungi, right? In contrast, you guys, what about the SAB? Only fungi. Right, it selects for fungi. It's selective for fungi. So my guess would be, you guys, but remember the microbes don't read the books. My guess would be you might have fewer colonies on your SAB plate, right? Might not always be true, but possibly. Now, you guys, the SAB plates, oh my gosh. Yeah, right? And you're breathing this stuff, you guys. You're breathing this stuff. Now, the thing is, on SAB, the fungi can grow really fast, right? And they will grow over their neighbors. So with the SAB plates, you guys, it is really important that you count colonies from the bottom. Because these great big colonies, often they will overwhelm their little neighbors. And you won't know that there's colonies underneath them unless you look from the bottom. So you guys, on this plate, it looks like, to me, 1, 2, 3, 4 have been overgrown. There's a great big guys, 5, 6, 7, 8, 9, 10, 11, 12. So you guys, on this SAB plate, it looks to me there's about 12 colonies. And again, you guys, count from the bottom, because these big fungal guys, they'll grow over their neighbors, and you won't know there's colonies underneath unless you look from the bottom. Okay? So you guys, are you proud of your immune systems? Just imagine, you know? That's amazing. Okay, so folks, first count total number of colonies. Okay, for your SAB 30, SAB 60, TSA 30, TSA 60. Okay? Yeah.
For this.
Oh my God, look at that, that's great. Wow, wait a minute. I think you've got a swarmer in here. Okay, I think you've got, which means they're so modal, they just swam over the whole surface. You know what, let's compare it to, yeah, I think you have a swarming, maybe a Proteus in there? See, this is uninoculated.
Some circles, but they don't look
Yeah,
any
yeah.
different.
Yeah. So, right, so for sure you've got one, and then you've got, oh yeah, here, right, 1, 2, 3, okay. My guess is you've got probably a Proteus in there, and that's what's covering the whole entire plate. So count that as 1, and then the additional circle. That's so cool. That's really nice. Yeah. So can you just, oh, to get the packet, okay. I might not have any more packets. You guys, let me stop the movie right now. Let me go look in my office to see if I have any more packets. If not, you guys, it is on Canvas, okay? But let me stop this.
I feel like